Welcome to Berries in the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
Hey, welcome everybody to In the Interim. And Don Barry has joined me again, and we have a really cool topic today to talk about in the interim. And that is the I-SPY 2 trial. Many people may have heard of the I-SPY 2 trial, so we're going to go back and remember a little bit of this trial. We'll walk through how it became, what it was, what happened in the trial. So, Don, you ready to tell us the story of I-SPY 2?
I sure am. Thanks, Scott. So I-SPY 2 has a history, and it revolves around Laura Esserman, of course. Laura Esserman was the PI. I was the co-PI of the trial, and this is back, we're talking 2007 and 2008. And she and I were part of a cooperative group called the CALGB, the Cancer and Leukemia Group B, which itself has a long history. And we were in the breast committee of that cooperative group. And we had designed trials and commiserated with each other about the lack of innovation and about what we wanted to do. And we designed a trial, I-SPY 1. You may wonder how the two came about, but I-SPY 1, where we were looking at neoadjuvant disease. Now, this is part of the story. In breast cancer, there were two kinds of categories of disease at the time. One was adjuvant, which was patients who had typically had high-risk disease, but were early patients, were newly diagnosed patients, and they had bad tumors, large tumors, tumors with positive lymph nodes, and were receiving a chemotherapy. Those with estrogen receptor status positive were receiving endocrine therapy, tamoxifen at that time. And what you would do is you would treat the patients after they had surgery. And after they had surgery means that for almost all of them, the tumor is gone because we took it out. And so what was the end point? The end point was when it comes back. Now, if you're into clinical trials, that when it comes back is a dicey thing. It sometimes takes a long time, and moreover, which is, of course, good for the patient. And moreover, it got better for the patient over time because these therapies worked. And that was a problem. The problem was that pharmaceutical companies and cooperative groups and the like, sponsors of these things and patient advocacy groups, it was difficult to run these trials. Just to touch the base, the other part is metastatic disease. Once it recurs, it becomes, let's say it recurs distantly in the lungs or the brain or the bone or something. Then it becomes metastatic disease. So that's a different category. We're talking about the so-called early breast cancer.
So let me see if the setting is typically when the diagnosis is made, the tumor's removed, and that's what adjuvant means. You go in, remove the tumor, and then you treat them with different therapies.
Yeah, the treating is called the adjuvant.
It's called the
It's
adjuvant. Okay.
adjuvant to the surgery.
To the surgery. And that trials ended up being extremely large because the endpoints took a long time. The treatments were reasonably effective. So clinical trials became incredibly onerous to run in this patient population in breast cancer.
Yeah, expensive, onerous. Nobody wanted to do it. What could we do? And what the CLGB said, and I was the faculty statistician and working with the committee and parcel of these decisions, we should try the neoadjuvant approach. Now, this was novel. It was led by something called the NSABP, another one of the cooperative groups that had led this, because the neoadjuvant approach is all you do is you exchange the surgery and the treatment. But that's a big deal because you're leaving the tumor in the body for like 6 months. But you're pummeling it with, lots of toxic therapies that kill lots of cells, including, of course, cancer cells. So it was a big deal, but it had a benefit. The benefit was that you got to see whether the tumor responded to the therapy that you gave them before you took it out. So you left it in, you watched it carefully. You saw whether the treatment you gave could be different in the clinical trial. Of course, it should be, because you're learning. And when you go in and do surgery after 6 months of therapy, you see whether or not the tumor is still there. And that, in the neoadjuvant approach, became the endpoint called pathologic complete response. You send the tissue to the pathologist, and pathologist can't find any tumor. Okay, so that was sort of a risk. I described it to my colleagues at the CLGB as betting the farm. This was novel. And we did studies in different categories of the disease, different biomarker categories of the disease. So I designed some of these trials, and meanwhile, Laura and I keep talking about what we're going to do. And we both wanted to do what I called the bandit approach, where you have multi-arm bandit, where you do lots of therapies and you try them on different patients and you see which patients work. And this, I had tried to do some of this back at MD Anderson, where I was and am in biostatistics department, and we designed trials in various diseases, and we Leos Pushtai, a faculty member there, now at Yale, and I tried to do this kind of thing where we look at various therapies and go to pharmaceutical companies and try to sell it. And it was a hard sell to have therapies from Eli Lilly, from Pfizer, from Merck, that you're comparing to a control, but in the same trial as you're comparing to each other, like because I had the data, I could look to see how the Eli Lilly was doing versus the Merck. And that's a dicey thing. So we failed, Leos and I failed. But we, in the context of the neoadjuvant approach, Laura and I mused about maybe we can do this in the neoadjuvant approach. And she is an amazing person. First of all, she never takes no for an answer. You say, you can't do that, Laura. Oh, yes, we can. And so who's going to fund this? We were told by various people, Anna Barker is a good friend of ours, and she was once the deputy director of the NCI, and she was the head of our committee in something called the FNIH, the Foundation for the National Institutes of Health, that it works with pharmaceutical companies, including the government, so it works across this. And she said, you'll never get this approved by the NCI. The CLGB was funded by the NCI, National Cancer Institute. And we would have to get the funding from the NCI if we were to do this at CLGB. And she told us, and others told us, and we knew actually, this would never happen. I had personally spent years trying to get innovations into the NCI with a little bit of success, but this would have been well beyond their ability to imagine this could happen. So we worked with the FDA. I remember the FDA. I remember Rick Pazder, still is the head of the oncology, telling me, but these are early patients. These are curable, and you're experimenting with them. How can you do that? And so I said, well, we're going to have a data monitoring committee made up of and a couple of great statisticians and a couple of great clinicians. And they're going to be meeting monthly and looking at the data, seeing how things are going and making sure that nothing. And he was satisfied with that. So they allowed us to do this study, but then how to get the funding. And the funding was through the Foundation for the NIH, the initial funder. But actually, Laura was responsible for getting most of the funding. She was a surgeon, who still is, and would treat patients, and they would tell her that she made them. They were alive because of her, and therefore, since I'm the CEO of this big corporation, we'll fund you. She got lots of funding that way. It was originally funded by the donations and philanthropy. As time went along and as we got more and more companies involved with their drugs, we passed the funding off on to them. So they had to pay to play.
So let's back up a little bit, make sure. So is I Spy 1 funded, or I Spy 1, you described this as a bit of a pilot, and from there you went out and were able to get this additional funding to bring in the first investigational arm?
Yes, exactly. I Spy 1 was funded by the government. It was funded by the NCI, and it was not randomized. Yep. Or adaptively randomized. It was looking at patients in the neoadjuvant setting because, these surgeons had to learn how to do this how this thing, not do surgery right away, but then come in and do surgery later. It was a new thing to surgeons. And I-SPY 1 had two endpoints. We were still looking at pathologic complete response, PCR. But we were interested in, could we predict PCR from an MRI that we give intermediate in time between the initial presentation and the initial therapies? After 3 weeks, for example, on a therapy, maybe there's an effect. And maybe that effect could predict not only path-CR, but also survival. So that's what I-SPY 1 was about. It was a kind of a registry, if you like, or it wasn't randomized. We weren't trying to learn about therapies. There was a specific therapy that patients got who met the eligibility criteria.
So interesting. People listening to this, you've described the neoadjuvant as a huge advantage to clinical trials and the ability to learn about the treatment. What do we know now about it for the patient benefit? There's no sense that this is better or worse for the patient that goes through neoadjuvant as opposed to adjuvant care?
I'm going to answer the question, but the only way I can answer the question is with hindsight.
Yeah.
So at the time, we thought that the benefit for the patient was this business about learning what benefits the tumor. When we would give therapies, different experimental therapies, and we do the MRI, sometimes we looked after 3 weeks and the tumor was gone. And that, is an obvious advantage to the patient that you can do something else if it hasn't gone. You could stop. If it's not affected at all, you might want to do something else. So it was getting the information that would help the individual patient, but it was also getting the information that would help in understanding which therapies are benefiting which patients. And then we could, emphasize those pairings, the right patient or the right therapy for the individual patient. And so it was this PCR that was the attraction for the clinical trialists, that perhaps you could get approval. And in fact, this has happened since. Perhaps you could get approval for a therapy that had a great benefit on the path-CR rate. Improves a 30% rate to 50%. Because patients who got complete responses did extremely well. And it didn't matter what the therapy was. It didn't matter what the biomarker characteristics were. If you were disease-free, as far as they could tell, at the time of surgery, and then follow them, they did extremely well for overall survival and event-free survival. Now, I mentioned, fast forward, we're going to have to go back to tell you about I-SPY 2, what it did. But fast forward with the neoadjuvant approach, what happened was there are patients who come out of the surgery, and as I said, when they're path-CR, they do extremely well. But if they don't have a path-CR, if they still have residual disease, they did poorly. The pharmaceutical companies latched onto this possibility and said, maybe we should treat those patients who have residual disease And to your point earlier, Scott, about the size of the trials, when we have residual disease, the event rate is a lot greater because they recur much sooner than and more likely than patients who get PCR. Patients who get a PCR don't need additional therapy. And moreover, in a clinical trial, it would be difficult to see that there's a benefit for the therapy because everybody lives. Not everybody, but most women. And so they did this, and they found, depending on the biomarker characteristics, that some therapies, immunotherapy, for example, worked extremely well for patients who were so-called HER2 negative. And the company would then go to the FDA and say, we've got this great therapy. Moreover, that's changing the course.
Yeah.
And it means that if you're a pharmaceutical company and you're using the neoadjuvant approach and you randomize patients to get your therapy versus the standard therapy, and you show that there's a PCR benefit, you're still not going to get approved by the FDA, because what happens is the patients who don't have a benefit, who have residual disease, are getting other therapy that's effective.
Yeah.
So it's confounding. And so the neoadjuvant approach has completely changed breast cancer treatment and approach. And it means that the original hope, it's sort of ironic, the original hope was to get a PCR, and now the benefit for the neoadjuvant approach is it really doesn't matter. You're just getting information about what therapy works, and then you give some other therapy, and they call it adjuvant, after the surgery. So that's a whole different story. That's not really part of I-SPY 2, but it's part of the breast cancer story.
Okay, so in the timeline of this, I-SPY 1 is proof of concept that you can do neoadjuvant. There's no investigational therapies. You can get MRIs. You're getting data on PCR. So meanwhile, if a company wants to do a phase 2 trial before I-SPY 2 in the adjuvant setting, it would be incredibly hard because disease-free survival after surgery is so very long. These trials would be huge and long. It's hard to do drug development. So you turn it upside down and you're doing neoadjuvant. So you're ready for I-SPY 2, which is intervention in the neoadjuvant space, and the funding. Are we ready to describe the I-SPY 2 trial?
Yes.
Okay. So, do you want to describe what this trial looks like? You want to do the bandit approach. You want multiple therapies simultaneously, but this is also about precision medicine in a sense.
Exactly. So the precision medicine aspect is breast cancer is probably the poster child of precision medicine. It has biomarkers that are extremely important: estrogen receptor status, progesterone receptor status, HER2 status, something called mammaprint, which is very similar to Oncotype DX, which is a 21 gene. Mammaprint is a 70 gene biomarker, polymarker.
So I Spy 2 classifies, you describe these, there's HER2 status, hormone receptor status, and mammaprint status. Each one is dichotomous for this. So women who come into I Spy 2 fit into a subgroup or a subtype, sorry. I Spy 2 created all kinds of new terminology. And there are 8 subtypes by the classification of that when a woman comes in, that their tumor is classified in eight different subtypes.
Right. And then we want to see which subtypes benefit from which therapies. But now if you what we want to do is tell the company, your therapy is beneficial in this subtype, but not in that subtype. Now, how can you do that? If there are 8 subtypes, there are 255 different combinations of those subtypes. And we can't do 255 different partitions of cancer. So we looked at what we call signatures, which are subsets of subtypes. And we looked at 10 that were the primary analyses. So every month we analyzed how well the various therapies in the trial were doing in these 10 different signatures. So one signature would be HER2 positive. That, as you described it, Scott, has 4 subtypes. But we're looking not at those individual subtypes, but at the all of them together. We also look at the HER2 negative. We look at the estrogen receptor positive and negative. We look at the estrogen receptor negative, HER2 negative, which is called triple negative disease. And that has 2 subtypes, namely mammaprint positive, mammaprint negative. That we're not advertising those subtypes as being an indication for your drug. It's the signatures which are indications for your drug. So that was a big deal to go to the FDA and say, we have these 10 different possible indications. And that in itself is a huge innovation, which, by the way, I Spy 2 is a phase 2 trial, and we're trying to ready it for phase 3. We've since then designed similar trials in GBM, glioblastoma, and pancreatic cancer that have a smaller number of subtypes because they have, different biomarker science associated with them. But that the FDA has agreed with looking at these multiple signatures in a registration trial. So it's pie in the sky, but we're eating the pie.
Mm. So a woman comes in and she belongs to one of these 8 subtypes and is randomized among control, and called 20% chance she goes to control, and the other 80% she could go to the various investigational therapies that are there at a time. So this is your randomized bandits in a way, that if there are 3 therapies in the trial right now, she gets randomized among the 3 therapies. And you probably need to describe the response adaptive randomization aspect, which is so critical to I Spy 2.
Yeah. So the bandit problem is that you have these multi-arms, and it's usually posed in the context of a single subtype, where you want to treat patients effectively in the trial, which is, I've been trying to blow up the notion that you can't learn, you're not treating patients in a clinical trial. You're learning about patients. And I say, why can't we do both? And we had done that. I've been writing about it for many years. And when I went to MD Anderson, we actually started to do it. So we had trials where we adaptively randomized. You get a randomization, but if that therapy is doing well for your subtype, if you're the patient, you get that therapy with a higher probability. And what it means is that patients in the trial, on average, have a higher PCR rate than a standard trial because they're more likely to get a therapy that's doing well. And moreover, you're not exposing those patients to a therapy that's not doing well for your subtype. And it means that patients in the trial do better. It also means that the overall patient population does better. And you get more information about the therapies that are doing well in the subtype / signature because they're getting it more often. So you get a bigger sample size because and faster. You learn faster, you learn better about where you really wanted to learn. You don't care how well it does in a therapy in a subtype that doesn't do very well with it. You don't use that for those patients. You use it only for those patients who it is doing well.
Okay, so when women come in, they're classified in their 8 groups, and the randomization probabilities for one subtype are different than another because the drugs are modeled as potentially having differential effect for those women.
Exactly.
And it might even be that they have no chance to get a particular therapy because it's not doing well, and a very high chance to get another one that's doing well for women like them.
Exactly.
From the drug side, the drug can focus on the women where it's having an effect and not randomize to those that they're not. What's amazing about this is you can't really do this. You can't do it very well if it's a single drug trial, because now you can't change the prevalence of various subsets without stopping enrolling them all together. It's almost undoable to do this precision unless you've got multiple things to give. Now you're doing these, I know you want to jump at this, but you're doing these adaptations and you're analyzing the data. The algorithms are being run and resetting the randomization weekly I think it varied during the time of I-SPY 2. I think initially it was actually daily, then weekly. Now, your endpoint is 6 months, and you're trying to learn about what works, and you don't want to wait 6 months, and so you're trying to accelerate the learning to allow all of these things you just described to do better. So how do you do that?
Thanks for the setup.
Yeah.
I mentioned earlier the MRI. We did MRIs in I-SPY 1 to learn about that, and we learned that it is predictive. What we like to do is to use all of the patients. We don't want to wait 6 months. In the context of waiting for disease-free survival or overall survival, 6 months is a short time. But in the context of I-SPY 2, 6 months is a long time. We get MRIs. We got MRIs. MRIs at 3 weeks and at 12 weeks. And we use that information to predict, is it going to be a PCR? Now, here is where I reported to the Data Safety Monitoring Board every month. And I explained to them the trial, and they kept learning about the trial. And after some period of time, they said to me, Don, you told us about MRI and how you use MRI at least 8 times. Could you tell us again? So it's not an easy concept for those people that are used to, looking at an endpoint and focusing on the endpoint. If you say, we're going to focus on the endpoint being MRI, first of all, nobody would accept that. It's not a surrogate for anything. But we looked at MRI not with the notion that what we're going to do is focus on MRI as the endpoint, but as an auxiliary endpoint, as a marker of how likely is it that it's going to be a PCR. So if there's no tumor that you can see on the MRI, that means there's a high probability it's going to be a PCR. So what we do is we do multiple imputation. We have this probability, and when we're doing the multiple imputation for all of the women in the trial who don't have 6 months, who don't have the result of surgery, we're predicting the result of surgery, but it's probabilistic. So there was a drug, pembrolizumab, which you've seen advertisements for Keytruda from Merck, which graduated. We call it graduate when we've learned what its signature is. When no patient had results at 6 months, and the way we could do that, we'll talk about the time machine in a minute, I'm sure. The way we could do that is we had the algorithm that was making the patient assignment and making decisions about graduation. The drug would graduate, no longer get patients, but we would continue follow-up. When only one patient had the result of surgery in triple-negative breast cancer. And how could we do that? It was because the algorithm was seeing that the therapy was melting the tumor away. And I went in and I looked at the data and said, how can it do this? And the answer was, in the 12 patients who had results at 12 weeks, 11 of them eventually had a PCR, and 11 of them it was clear that they were going to do extremely well. And at the time, I think one patient had been through 6 months, and the model predicted something like a 63% PCR rate for the arm. And with one patient, and by the time all the patients got through, it stopped for graduation at some point. It almost nailed exactly the PCR rate based on the MRIs. It was exactly the same, same as the MRI. But now there's another aspect to this. How could it do this 60% and get it right with only one patient? The answer is the time machine and the controls.
So hang on. We're going to be adaptive here. So for the first time, we're going to call this part A of the podcast. And we're going to make people have to tune in for part B of the podcast and learn about the time machine. And thank you all for joining, and join our next episode and learn about the time machine.
And other things as well. Predictive probabilities, for example. which are usually important in being adaptive.
All right. So thank you.
Thank you.